Welcome biologists, Miss Ray here. We will be learning all about patterns of inheritance in Unit 5, Lesson 2 notes. Last lesson we learned all about Mendel's discoveries as he came up with his 3 laws to explain what he was seeing in his garden. But as we've learned more over the centuries about genetics, we now know that inheritance can be a lot more complicated than we think. Have your packets ready and let's get ready to write.
So in lesson 1, we learned that Mendel had created these 3 laws: law of dominance, law of segregation, and law of independent assortment. And we're going to focus on law of dominance at first. So in Mendelian inheritance, which are inheritance that follows that law of dominance that Mendel created, we know that there's something called complete dominance. And that's what we've actually been practicing, where when we see that big letter, that uppercase letter, that means that it's dominant and masks the smaller case or lowercase letter. So complete dominance is a dominant allele completely masking a recessive allele in heterozygous genotypes. We use the same letters, just uppercase and lowercase, to designate whether something is dominant or recessive. And letters that are usually selected based off of the dominant trait. So for example, if red is the dominant trait, then you usually use R, or if white is the dominant trait, then you're going to use W. If tall is the dominant trait, then you're going to use T, things like that. Now, you see all these examples in these images. For example, you have the big R, little r. Because it's heterozygous, it follows the dominant trait, which is red. So it's going to be red. And that the only time you'll ever see a recessive phenotype is going to be when you have 2 small letters, because there's no dominant that's masking it. Now let's practice a little bit. Again, this is a throwback to lesson 1. You should be able to do this. So complete the genotypes and then fill out the phenotypes based off of what you know. All right, so you have, if you have these answers, you did them correctly. You can see that our big B, little b will give us black fur. Our little b will finally allow us to see that recessive allele, white fur. With our dimples example, big B has dimples, of course, but big D, little d has dimples also because of that dominant allele. And the only time we see no dimples would be if the organism had little d, 2 recessives. Now here comes the catch. Yes, we have this law of dominance and we have these other 2 laws, but sometimes organisms do not follow the law of dominance. This is going to be when you see organisms that are a blend of color, or have multicolored, or splotches or spotted or things like that. And these follow something we call non-Mendelian patterns of inheritance. And there's multiple ones, but we're going to hyper focus on incomplete dominance and codominance. Now, the rule of thumb for all of these, whether it's codominance, incomplete dominance, or complete dominance, is look at the heterozygous phenotype. If you have, again, a blend, it's incomplete. If you actually see the dominant trait, then it's complete dominance. If you see a mixture of both, and you see both colors or both phenotypes, then it's codominance. We're going to practice this now. So we're going to focus on incomplete dominance first. Think of the word incomplete dominance. You have multiple alleles that are dominant, but they're not dominant over the other. What that actually gives us when we have them mixed together is a blend. It gives us this new 3rd phenotype that looks different from the other 2. For example, in the flowers, we took red and white, and if you made offspring from those, the offspring would be pink. So not fully red, not fully white, but a pure mixture of both. Incomplete dominance is that heterozygous individuals present a blend of 2 phenotypes. Neither trait is dominant to the other, so they make a blend or mix of both that occur in heterozygous. Different letters are used to represent each allele. And again, the heterozygous in this pattern is a blend or mix. In the case of the rabbits, you have a black parent and you have a white parent, and the individual offspring ends up being a blended gray rabbit because, again, black and white makes gray. So again, if you see this new third color popping in, that is, boom, it's going to be incomplete dominant. So incomplete is a blend. Incomplete is a mix. Make sure that those two words, again, are synonymous with incomplete dominance. Now let's practice it a little bit. I've helped you out with the first one. So again, homozygous, we're not using dominant or recessive anymore because there is no recessive. So you have same letters versus not same letters. So same letters with big B, you get black fur. Heterozygous with a B and a W, you get that blend, which is going to be gray. Now, WW, you're going to have white. Now for the next one, I want you to finish this chart. I want you to think about what would happen if you mixed curly hair and straight hair to form this new phenotype. All right, if you got these, so curly hair for CC, wavy hair for CS, and SS straight hair. So when you, again, blend together curly and straight, you get wavy. I'd like to say that this is the only one we have, but we have this other one. We have something called co-dominance. Now, co means with. That means that it's dominant together. Like co-worker, you work together. So co-dominance is the heterozygous individuals that present traits from both alleles. You see them both. Now, again, we're using different letters. So different letters are used to represent each allele. And heterozygous in this pattern is going to be spotted or equally but individually visible. So let's take a look at some of these examples that we see. We see these cows, all right? So we've seen spotted black and white cows, all right? That would be an example of codominance. If you look at the chickens, if you look very closely, you see both gray and you see white. It's not just one or the other. That's usually called a speckled appearance. The fish, you can see where you have both red and blue in the offspring. And in the flowers, you see that you have both red and white exhibited in the offspring. Now, one thing that you can get confusing is you want to say, oh, well, flowers, oh, I saw flowers that are pink. That means that all flowers exhibit incomplete dominance. Or I see these spotted or splotched flowers. So all flowers are codominant. And that's not the case. You have to take it species by species and example by example to see what kind of pattern that specific organism is going to exhibit. Now, let's practice this. Again, do the genotypes first. See what needs to be filled in next, and then try your hand at filling out the phenotype. Were these the answers that you got? So again, when we have different letters, so we're not seeing big B, little b, or big W, little w, or big R, little r. We are seeing different letters representing the dominant alleles. Again, you don't see homozygous dominant because you don't have one that's dominant over the other. So we just use the term homozygous, which means same letter, and heterozygous, which means different. Now, with the fur color, in this case, you have black fur for big B. B and W make black and white fur, and WW will make white fur. In petal color, this will be RR, red petals, RW, red and white petals, and WW, which will be white petals. Now, we're now going to link this to Punnett squares. Note that this year you're only going to have to read Punnett squares rather than create them, but we have to talk about probability. So probability is a measure of how likely something will happen. We use Punnett squares to just predict and say, okay, you have a 25% or you have a 50% possibility of the offspring looking like this or having this traits. It's good to sort of understand, but the reason why we're really just creating them is it's a good visibility of visual way of understanding heredity, but we don't really use them in the real world very much. So the action of actually having to read to create them is just not a very useful trait. So what you do is you count how many of a possible outcome that you're looking for, and then you put it over the total. And when you divide that, you're going to get a percentage. Now, the percentages is really easy. If you have 1/4, that's going to be 25%. If you have 2/4, you're going to have 50%. If it's 3/4, that's 75%. 4/4, that's 100%, and 0/4 is 0%. Those are pretty much all the percentages that you're using, and you should have those memorized. Now, in our example, if you had 1/4, that would give you a ratio of 1/4, which would be 25%. Uh, Look at the Punnett square that's on this image. This is a different one than our example. How many would be green? So how many of the 4 would actually be green? You might have noticed that there are 2 that will be green, and it's out of 4 total offspring. So 2/4 would be 50%. If I asked you how many would be yellow, you're going to notice that it's also 2/4, which would be 50%. And the nice thing is that you can always add those percentages up at the end. They should equal 100, and that lets you know that you did your math right. Now, Punnett squares, these are tools to help make predictions about the outcomes of a genetic cross. We use them again, we use probability to predict possible genotypes and phenotypes and the combinations of offspring. It is not a guarantee that this is the offspring that an organism will have. It is just a possible combination. Now, the best way to actually do this is to practice. So you have 4 practice problems in your packet. We're going to do the first one together, and then you're going to do the other 3. Your teacher will be checking to make sure that you do them. Try them out. Again, if you don't practice them, this is not going to make sense. So let's look at the first one. In pea plants, the allele for tall, capital T, is dominant to short, lowercase t. If a heterozygous tall pea plant is crossed with a homozygous recessive short pea plant, what is the probability the offspring will be tall, short? What kind of pattern of inheritance is this, and how do you know? So the parent cross is already given to you, but we have to figure out, before we do anything else, we need to figure out what type of inheritance. Is this complete, incomplete, codominance? So look at the question. We're going to focus on the heterozygous. We were told that the heterozygous is tall. Now remember, we also have mentioned that we have dominant and we have dominant. Something allele that is dominant over the other one, which means that we have a recessive one too. That's going to be our big hint that we are dealing with complete dominance. And how you explain that would be complete dominance because you have a dominant and recessive allele, and the heterozygous demonstrates the dominant allele. So when you look at this square, you'll notice that, big T, little t is going to give you a tall. You have little t, that'll give you a short. Big T, little t, that'll give you tall. Little t, that'll give you short. So when we go through and we start making our ratios, big T, well, there's actually not any of those in there. So we're going to put 0/4, and that's going to give us 0%. Big T, little t, we have 2 of those out of the 4. So 2/4, that's going to give us 50%. And little t, we have 2 of those, and that's going to give us 50%. Now let's check our answers. Make sure that this is what you have. I also find it nice to go through and identify in the Punnett square actually what something is. So it wouldn't be harmful for you to write within the box, big T, little t is tall, and little t is short. It'll help you with not making mistakes. Now you're going to complete the other 3 doing the exact same thing. Sometimes you'll have more information given to you, sometimes it'll be less, but you should be able to identify. Remember, look for the heterozygous phenotypes to identify which type of pattern of inheritance you're using. I know this is tough, but you guys can do it. I believe in you. Have a great day.